Let me just get a laser pointer. All right, well, looks like this is going to work. So I'm sure you're all familiar somewhat with Lyme disease. This is a infectious disease that is transmitted, of course, by ticks carrying Borrelia burgdorferi, which in order to confuse us is now called Borrelia burgdorferi. Taxonomists are probably bored, and so periodically they rename common species. And so the estimated number of cases is now close to 500,000, according to CDC. In the US alone, that's a big number. If you get something like a bullseye rash or a rash and suspect that this may be a result of a tick bite, you're treated with typically doxycycline, and Lyme goes away. Unfortunately, and the reason I am interested in the subject is that about 10% of people who've been diagnosed in time and properly treated develop post-treatment Lyme disease syndrome, according to how CDC calls it. And now I know that patients do not like this term. They prefer the term chronic Lyme. Doctors hate the term chronic Lyme. They think that it doesn't exist. So now there's been a trend to rename it into long Lyme, by analogy with long COVID. There's been a recent publication in Lancet comparing symptomology of this chronic ailment after a tick bite to symptomology of long COVID, and they are very similar. It is, muscle pain, foggy mind, fatigue, stuff like that. That also shows up in a number of autoimmune diseases. So, as you probably know, the existence of this post-acute Lyme stage has been highly controversial. I would say that mainstream doctors probably do not believe that it exists. Now, there's been recently a very nice paper that's coming out this year. It's already available online, from John Alcott from Johns Hopkins, who's my collaborator, who did a very careful study comparing symptomology of these chronic symptoms of patients who had Lyme with such symptoms showing up in the general population. And indeed, in the general population, we have about 4% of people who, for reasons that we do not understand, will complain of this set of symptoms. But if you've been bitten by a tick and had Lyme, then that percentage goes up from 4 to 13. So again, we're about back to 10%, where it's sort of people who've been studying this phenomenon felt that that is about the correct number. And I think that that paper hopefully will put to rest a lot of this controversy. But, you never know. And that probably, apart from just speaking of strictly scientific subjects, I will also take the opportunity, since I'm talking to a lay audience, or at least not the experts in this subject, I will tell you of things that may be of interest to you that are connected to this theme. When I think about this controversy, I'm reminded of the controversy, 19th century controversy, about the role of pain and anesthesia for surgeons. So in the late 19th century, doctors from MGH invented anesthesia using ether for general anesthesia for operations. Now, before ether, surgeons had no choice but, to cut a person open. And of course, that caused horrible pain. So the doctors came up with a theory that pain helps heal wounds. And so then everybody should feel better because pain is good for you. So then ether becomes available. And for a very long time, a very large portion of surgeons refused to use ether because they believed based on no evidence that pain is good for you. All right, so I take doctors' reluctance to accept certain concepts with a grain of salt. And again, doctors, unfortunately, just as patients, are in a very unpleasant situation when a patient tells them that they have chronic Lyme and they expect to be treated. And we do not have treatments for this disease. So it's psychologically, I understand, it's considerably more convenient to say that this disease does not exist. And therefore, you don't have to treat it. All right. So much about human psychology. So let's get back to science. Now, for a number of autoimmune diseases, there's been a distinct connection made between the composition of our gut microbiome and disease. And there are a number of companies now that are trying to fix the microbiome for patients that suffer from, for example, lupus and other autoimmune diseases. There's no question that our microbiome is in contact with and affects the immune system. And that in turn, if it gets out of sync, may cause symptoms that are very similar to long Lyme or long COVID. So with that in mind, in collaboration with John Aukot, who I already mentioned, we decided to have a look at the composition of the microbiome of patients who suffer from these chronic symptoms. And so there's a very nicely evaluated cohort, not a huge one, but a reasonably sized, 87 patients. This is a Johns Hopkins cohort. Those patients donated their stool, which we analyzed by whole genome sequencing and some other methods. And so we know the composition by species of the microbiome of these patients, of each of these patients. And we plot that and compare that with the composition of microbiome from our controls. And we have two controls. One is this control, ICU, patients from intensive care units who are heavily treated with antibiotics. And they, of course, they group together, and that's rather clear grouping. And then there's healthy here in blue. They also group together. And now these are PTLDs that group in a separate group of their own. We call this the microbiome signature, and that we published not so long ago, 2 years ago. Okay, so this signature, we figured, can actually be used as a diagnostic tool. So here, if you look at this plot, which was produced with the machine learning algorithm, and I cannot tell you that I'm an expert in machine learning. That was done by Rob Knight, my other collaborator. But essentially, in this plot, if the deviation of the microbiome you're interested in from normal is 0, if it's essentially normal, then you get this diagonal. If it deviates very strongly, then you get this square. So this is almost a square. So the result of that is we can determine correctly in 82% of cases that a person has long Lyme simply by looking at that person's microbiome composition. And now a company, FlightPath, has taken this approach and is evaluating the signature on a considerably larger number of patients. Now, another thing that we did is we compared how this determination by microbiome signature compares to other cases where people have tried to do that. And it comes up on top by comparison to other cases, including such distinct as, for example, Crohn's disease and colitis. Okay, so what is it exactly that is happening in the microbiome of these patients? So we had a careful look, and we were looking at drivers at those microorganisms who changed the most. There are lots and lots of changes, but here we're looking at those that changed the most. Well, one thing that becomes rather obvious is that there is an expansion of this creature, Blautia, that is suspected to be pro-inflammatory. We don't know anything good about it, so maybe this is, as we refer to, as a pathobion. So not necessarily distinctly causing a disease like cholera causes cholera, right? We cannot draw that simple one-to-one connection, but maybe this is, not so good to have in your gut. And at the same time, what we see is a decrease in the abundance of Bacteroides. So these patients have an expansion of Blautia and a decrease in Bacteroides. Bacteroides are symbionts. So this is a very large and dominant group of symbiotic bacteria, and they do a lot of useful things in our gut. One useful thing that they do is they produce short-chain fatty acids, so simple stuff like, acetate or propionic acid. And the cells of our epithelium, gut epithelium, they use these short-chain fatty acids as food. They get most of their food directly from the gut, which is probably not surprising since they're facing the gut. So these bacteria essentially, feed our gut lining and contribute to maintaining its health. But they do another interesting thing that my lab discovered several years ago. Bacteroides, interestingly, and quite somewhat unexpectedly, produce GABA, gamma-aminobutyric acid. It's one of our major neurotransmitters. If you don't have enough GABA, you can get depression and anxiety. And actually, GABA agonists are, standard medication to treat depression. So now there's my discovered that Phil Stranowitz, he started the company Holobiome in Cambridge, and their goal is to package Bacteroides in a pill. Colonize the gut of those people who don't have enough of them, and hopefully improve their mood, apart from other things. And mood disturbance is one of the hallmarks, actually, of chronic Lyme as well. Another thing that's happening, if you look here at the right, is the bloom of Enterobacteriaceae. In some patients, you see 100%, they just took over the microbiome. That was quite unexpected. When we talk about dysbiosis, and this is a word that everybody, I'm sure, is familiar with, that usually means a bloom of Enterobacteriaceae, so bacteria like common E. coli, that is a contaminant in food and drinking water, etc. And its relatives. And these are pro-inflammatory bacteria. And if you have dysbiosis at any age, or let's say elderly people, especially in assisted living conditions, they tend to have a lot of these Enterobacteriaceae in a generally impoverished microbiome. All right. So this is what we found. And one conclusion from this is that it would be nice to fix the microbiome of patients with long Lyme, and that's one of the things that my lab is working on. So from that we arrive at this hypothesis of what is it that causes long Lyme. So you have an infection with Borrelia burgdorferi, which affects the immune system. Of course it does. It's a pathogen. In turn, the immune system affects the microbiome, causes an alteration, which in turn feeds back to wreck the immune system, and you get long COVID. But there's another component which I think is maybe very important, and that is paradoxically treatment with broad-spectrum antibiotics. So of course you have to treat with antibiotics, and the only ones available currently to treat Lyme are broad spectrum. So they will kill Borrelia burgdorferi, and they'll kill pretty much lots of other things, useful and symbiotic bacteria in your gut. So Borrelia burgdorferi affects unfavorably your microbiome, and then you come in with broad spectrum antibiotics, and they finish the job. That's the way I see it. So then, apart from fixing the microbiome, what I think would be very useful to have in our arsenal of treating acute Lyme disease, first of all, acute Lyme disease, is a selective antibiotic, an antibiotic that is going to kill only Lyme, Borrelia, burgdorferi, is not going to affect the microbiome and will be much safer from any perspective, but also very specifically will prevent the development of PTLDS if this hypothesis is correct. And that's what I will tell you about now. But let me first very briefly introduce to you how we discover new antibiotics, and that's really what my center works on. We are an antibiotic discovery center, and over the past, I don't know, decade, we discovered a number of pretty interesting antibiotics that are now in development. And so typically you take a sample of soil. Soil is where you get a very diverse community of microorganisms. They fight with each other, and they fight each other with antibiotics. So then we borrow these antibiotics and introduce them into clinical practice. How do we borrow them? Well, we isolate the bacteria. Then we place them here. You see there are three colonies of different bacteria placed on a petri dish, which is overlaid with this lawn of a test pathogen. In this case, it's Staph aureus. And you see that one of these three colonies makes a zone of inhibition. So it kills Staph aureus around it, which means that it's making an antibiotic. And then it's a question of taking this colony, growing it up in a larger flask or in a fermenter, isolating the active principle, determining its structure, figuring out if it's novel or not, and then testing, first of all, against human cells to see if it's not toxic to humans, and then test eventually in an animal model to see if it clears the infection in animal. If it does, and it passes lots of additional toxicity tests, then you go into clinical trials and you get an approval. FDA approves a new drug. That's sort of, in a nutshell, how it happens. Now, there's a big problem or an enormous bottleneck in this process, and it is this step, which we call chemical dereplication, trying to figure out whether you have a novel compound or not. It's not like we don't know how to do that. Of course, we do. And we can fairly rapidly, within a month, I would say, we can go from this zone of inhibition to a pure compound with an understanding of its structure. And I think it's terrific that modern science can do that in about a month. The problem is that the majority of bacteria like to make stuff that from our perspective is junk. It is detergents, DNA intercalators, redox agents, things that are toxic to us, just like they are toxic to neighboring bacteria, right? So that's sort of one enormous background that you get. And then another also big one, maybe not as enormous, but big one, are known antibiotics. You know, people have been studying antibiotics since the 1940s, so we discovered a very large number of them. Most of them are not useful, but they are there, and you're going to repeatedly re-isolate them. There is not a general solution yet to this problem, although my lab is working on a general solution to this problem, which I think I may have. But from this general approach, I will tell you what we've been doing in searching for selective compounds against Borrelia burgdorferi. But let me, again, since this is not strictly speaking my professional science talk, I decided that I will tell you something you may find interesting, and that is where ideas come from. I cannot say about everything. I have no idea where ideas of other people come from, but I know where my ideas come from, right? And I decided I'll share that with you. Of course, we never write about that in a paper, in a publication, right? I never say, oh, this is how I came up with this idea. But here I'm going to sort of confess to you how this happens. So I could also tell you, again, speaking about myself and probably about most of my students, that I know what is the main obstacle for invention. Of course, we need to invent. We have a problem. We have no idea how to solve it. You need to come up with something new. And there's one thing that is highly detrimental to invention, to getting new ideas, and that is our common sense. Because the common sense is always going to tell you, don't go in that direction because that's not productive, it's not going to work, nothing will happen, and so don't even think about it, right? And that sort of keeps you inside the box, your common sense, enemy of invention. There have been two successful approaches to eliminate common sense, and I'll tell you about these approaches. So one approach invented by Alex Osborne, who came up with formal brainstorming. Now, of course, everybody heard the term brainstorming, but formal brainstorming is different. Alex Osborne, American advertiser, worked in advertising, and he discovered that in the field of advertising, there's not enough creativity. And he suspected that common sense is to be blamed. He came up with a way of eliminating common sense. So he said that let's have a session, a discussion amongst a group of people who are going to solve a problem or propose something new. And we'll have two rules. First, we will only have positive comments in the first part of our discussion. And then once we come up with something terrific, then we'll have an analytical dissection to see if that's realistic. That very simple proposition turned out it's now a field of psychology, formal brainstorming. Professors in academia do not use it and do not know about it for whatever reason. But I learned about this in the industry. Industry uses formal brainstorming. Usually a professional psychologist runs these sessions, and I now adopt it in my lab. I have my center retreat once a year. We go somewhere for 2 days and we do formal brainstorming where anything that you propose is met with enthusiasm. So then everybody relaxes, everybody turns on their creative abilities, and sort of harmony happens, and this is really terrific, and we come up with new interesting things. And then there's another thing that I use personally, also tell my students about that, of course, but practically everything that I invented comes from this second approach. It was introduced by Genrikh Altshuler. Altshuler was a Russian engineer who published his first work in 1956, also a while ago, not known to the West, then became known, when people started immigrating from the Soviet Union. And now there's a community called TRIZ. It's an acronym for an unpronounceable Russian name for this. Now you can check it up on Wikipedia. There's a reasonable description of what there is, and there's groups and societies of people. And a company in Boston, for example, a company of inventors that use very elaborate rules of this approach to invent stuff for blue chip companies like Intel when they get stuck. But that's the world of engineering. Again, biologists don't know about that. But I, by accident, read Altshuler's book when I was a student, got captivated. And what captivated me is he has sort of lots of things, but many of his things, I think, if you do science for a long time, you intuitively come up with those tricks. But one thing very hard to come up with. He says that formulate an ideal result without considering technical feasibility. Right. So this is, I think, an important breakthrough. Completely ignore technical feasibility. Only ask, what is it ideally that you want? And then worry about whether this is practical. So at the first stage, you're eliminating your common sense. That's very important. So here's our problem. And here's the application of Schroeders Principle. Here's a Petri dish. Remember, I showed you a Petri dish. We put a colony on that petri dish, and let's say that this is a petri dish with a lawn of Borrelia burgdorferi on it. We put a soil bacteria here, there's a zone of inhibition. So what would be the ideal result? Well, the ideal result would be to look at this petri dish and say, okay, this zone of inhibition contains a novel compound, and we know that without doing any chemistry. And this novel compound is non-toxic to humans. And we know that without doing any tests with human cells or mammals. This proposition that I just submitted to you sounds absurd, which is also very important. Schroeder had this to say: if you formulated an ideal result and it sounds absurd to you, you are onto something. All right. So thinking about this proposition, this is what we came up with. Now, we are looking for a compound that's going to be selective against Borrelia burgdorferi. Well, so how about we take this colony that's producing something and killing Borrelia, and we'll also place it on a petri dish overlaid with another pathogen, with Staph aureus. And let's say it produces a zone of inhibition here as well. Well, then we're not interested. This is clearly a broad spectrum antibiotic. That's not what we're looking for. But if another organism, microorganism, produces a zone of inhibition on a Borrelia plate, but not on a Staph aureus plate, well, then we have a selective compound. Of course, this also contains a bet. We are betting that Mother Nature would bother to make a compound that is selective against spirochetes. Borrelia burgdorferi is a spirochete. They're soil spirochetes. And so that was our reason. But if you can find such a compound, then there's some interesting conclusions that we can draw immediately by just sort of looking at what I'm showing to you right now. You see, we don't know of any natural compounds or any compounds that are selective against spirochetes. So then whatever we discovered is new by definition, novel, right? And we've done no chemistry. And then if it's a selective, this compound, against a particular type of bacteria, well, then it does something specific to that particular bacteria. It's not going to harm human cells because, of course, human cells are very distinct from that particular bacteria. So again, not testing human cells, we know there's going to be non-toxic, right? So we were happy with that guess and went on to check it out. So we had a collection of bacteria from soil, you ferment them and indeed test against Borrelia and then parallel against Staph. Find this with something like this that we would say is a hit. After isolation structure determination, we came up with this structure of a compound that I was pretty confident is novel because of the logic that I just laid out to you. I plug this compound into a database that looks for matches with known compounds. And I was confident that there is going to be no match. But to my surprise, there was a perfect match with Hydramomycin A. Hydramomycin A discovered in 1953, a very long time ago. That I thought was very strange. First of all, I never heard of this Hydramomycin A. What is this compound? And secondly, I was confident that Hydramomycin A was not known to be selective against spirochetes. So we tested it against a bunch of bacteria. These numbers may not tell you much, but believe me that these are very high. This is very poor activity. It has also poor activity against our gut symbionts, so a lousy antibiotic, I would say, because it doesn't hit any of the pathogens. But if you test it against spirochetes, you see how these numbers jump down. This is minimal inhibitory concentration, the minimum amount of antibiotic that you need to add to kill Borrelia burgdorferi is 0.2 microgram per ml, tiny, tiny amount, very potent. Very also potent against Treponema pallidum that causes syphilis, against all Borrelia. And we tested and against, here's an environmental spirochete, for example, also pretty potent. So here we had on our hands a selective antibiotic acting against spirochetes. And since nobody tested them, it was just an old abandoned antibiotic that nobody cared about. But we now care about it, and we decided to look at it more closely. All right. So one thing was known about this compound, its principal mechanism of action was determined. This is an inhibitor of protein synthesis. So it inhibits the ribosome that is in charge of synthesizing proteins. This compound acts against protein synthesis in Borrelia exactly the same way as in E. coli, where that mechanism was initially determined. So while we know how it kills bacteria, that doesn't tell us anything at all why it is selective against Borrelia burgdorferi, because it shouldn't be based on its mechanism of action. Mechanism of action is the same. So then we decided, well, maybe what is happening is that maybe this compound is selectively accumulated by Borrelia burgdorferi. And that's exactly what happens. It is accumulated very rapidly by Borrelia burgdorferi and not so much by E. coli. So that's why the huge difference in susceptibility of these two organisms. So this is what we found. We figured out the mechanism of that selective uptake. So this compound Hydramomycin A is a mimic. It's a very clever mimic. It looks a little bit like common nutrients that spirochetes require, and these are nucleosides, building blocks of RNA and DNA. Spirochetes don't make their own, so they have to rely on whatever nucleosides are present in the environment. And they accumulate them with the help of this rather complex transport system that brings these nucleosides inside the cell. This transport system is tricked by Hydramomycin A. And it takes up Hydramomycin A and accumulates in the cell. And then, of course, it hits the ribosome, bacterial ribosome, and stops protein synthesis and kills. That's where the selectivity comes from. Okay, so then, of course, we checked out this compound for efficacy in the mouse model of Lyme disease. And you see what is by oral treatment with a fairly modest dose, comparable to what you use, actually somewhat better to what you use with ceftriaxone, a very potent it's a medication used in sort of difficult cases of Lyme disease. It completely eradicates, we cannot find a single live cell in that mouse. There's another interesting thing that we observed. We also decided to test Hydramomycin A against Peromyscus mice. So these are white-footed mice that are the primary reservoir of the Borrelia burgdorferi in nature. There was an interesting study done again by CDC about 15 years ago, where on a small island, they decided to test whether they can eradicate Lyme disease. So they spread doxycycline in baits so that mice ate the baits, cleared themselves of Borrelia burgdorferi, and Lyme disease disappeared from that environment. Right. So we now, that's all lovely, but you're not going to spread doxycycline over the United States or over any large territory, because it's an important human antibiotic. It's broad spectrum. It will kill bacteria indiscriminately. But for Hydramomycin A, which is highly selective against spirochetes, that's a different story. So we got a grant from the NIH. And are gearing up for our first field trial this summer to see if we can eradicate Lyme disease from the environment. Because of course, ultimately, while treating is nice, ultimately you want to get rid of this disease. And I think this is one of a very few infectious diseases that you can truly eradicate and get rid of. And the last thing I want to show you is what is indeed happening with the microbiome. Because remember, our main motivation for this antibiotic discovery project was to see what happens with the microbiome, whether Hydramomycin A will damage the microbiome as broad-spectrum antibiotics do. So here's an experiment with mice. Let's have a look at the control. So this is amoxicillin, and this is ceftriaxone, commonly used antibiotics. And you see that there's an enormous expansion of Enterococcus, which is a pathobiont. Essentially, it takes over the microbiome in the presence of these antibiotics. And in case of Hydramomycin, there are a little bit of changes here and there, but nothing really dramatic is happening. So I would say this sort of a very mild effect, if any. So that gives us hope that this compound has legs, and we are currently in extensive preclinical evaluation of this compound, and hopefully this compound is headed to the clinic. I'm very encouraged by the fact that we find absolutely no toxicity with Hydramomycin A. That's the usual bane of any new medication, and antibiotics especially usually look for a therapeutic window, a difference between a toxic effect and efficacious effect. Here, we don't know what the window is. We cannot determine it. We know that it's big. That's the only thing that we know. Okay, so with that, I would like to thank my team, my collaborators, and of course, organizations that support our work. This primarily came from National Institutes of Health and Stephen and Alexandra Cohen Foundation that supports work in Lyme disease. And this is my team. Thank you. I will stop sharing now.
Wow, thank you, Dr. Lewis, so much. Great. So for the people, participants, if you have questions, feel free to put it in the Q&A and the chat, and I will be reading off a few. Before we see a few coming in, I had a quick question. So you mentioned, in addition to like kind of working towards clinical trials on human, I'm assuming you meant on human or for treatment. You were also saying that there were field trials that were going to be happening this season or in the near future. Can you tell us about
this coming season? Yes.
And where would those be? I'm just curious.
That I'm not going to tell you, Sarah.
Oh, okay.
I'm not going to answer.
I didn't think it was a secret. But
And the reason, Sarah, I'm not going to tell you is because I want that field trial to actually take place.
That is very fair. I know a lot of us that are on Nantucket, you know, we've had lots of different researchers come out to study the ticks here, the tick-borne diseases, the different vectors. And so it's really interesting hearing from different researchers coming at it from different directions. So treatment of people and people that have been affected, but it's really interesting to me that you can use the same treatment, basically to both treat the person that's already infected as well as, you know, the vectors along the way.
Right.
All right, so we have thank you for working on this, as some people have said. One question is, what do you think about the IGeneX blood test for determining the level of Lyme's and Bartonella? Is there something else that works well? And thoughts on long-term treatment with doxy and azithromycin? Sorry.
So there are two parts to that question, and the second part is I didn't quite get it. Could you repeat the second part? Does it?
So right, the first part is about the blood test determining the level of Lyme's, and then is there something else that works well? And then their thoughts were, and then they wanted your thoughts on the long-term treatment with doxycycline and.
Got it.
Yeah. Okay.
All right. So I'm not an expert in diagnostics. It's a separate field, and this is, you're talking, of course, about diagnostic of acute disease. What I do know is that there are a number of experimental diagnostics coming up that I think will probably be much better than existing diagnostics. So one approach that is currently being evaluated is analysis of not of specific antibodies, which tend to come and go, sometimes causing false positives and false negatives, but a more detailed approach where you simultaneously measure expression of all genes from blood. Enormous amount of data, and so their small fluctuations or variations are canceled out. So I think that approach is probably the best promising one. Now, as to long-term treatment, I understand that you are probably asking about patients who have long Lyme or chronic Lyme, and how should they be treated with antibiotics, et cetera. Now, there have been clinical trials of patients with chronic Lyme with antibiotics, and those trials have come up primarily negative. Those trials are imperfect from the way I see it, because they were performed, of course, with broad spectrum antibiotics. These are the only ones that we had. So on the one hand, so let's say there's some residual pathogen hiding somewhere, right? That's the premise of such treatment and trials. Well, if it's hiding somewhere and you hit it with a broad spectrum antibiotic, you're also damaging your microbiome at the same time. And if you get a negative result, the way I see it doesn't mean anything, because that's not the correct experiment to do. The correct experiment or clinical trial for patients where they may or may not have residual pathogen would be with something like Hydramomycin A that selectively eliminates Borrelia burgdorferi. If such patients get better, that will tell us that they were suffering from residual pathogen, right? So that's one of the things that we sort of looking forward would really like to address.
So I'm going to put a few questions together. There's some questions about the timeline for these clinical trials in humans. So, kind of where is that at for timeline? And then even with the clinical trials, what's the timeline further for if it proves to be successful for a drug that would be available?
Yeah. So, so we're now in preclinical investigation. This should take, so we're kicking off. One of the things that needs to be done is large scale production. And this we're kicking off this spring now with an industrial company that does that. The whole process with production included should take 18 months. So that's, once that's done, if everything went well, you get then approval for clinical trial. So the clinical trial typically has 3 phases, 1, 2, and 3. Clinical trial 1, which looks at safety, just the lack of toxicity in humans. Here, I think everything should be fine, and this would be relatively short, a couple of months. Then there's phase 2, which looks at efficacy, so actually treating patients with acute Lyme disease. So there would be, a group that's treated with doxycycline and a group that's treated with Hydramomycin A, right? And there will be a comparison. That takes about a year. And then there is sort of bifurcation at that decision point after phase 2. So for at least some antibiotics, FDA will allow, especially for something principally novel, will allow introduction into clinical practice after successful phase 2. That doesn't happen for other medication, apart from compassionate use in cancer. If FDA allows that, then that'll be a short timeline. If not, then you go into phase 3, and that's another 2-3 years, a large number of patients, et cetera. Then after that, well, production is already has already been done, right? Before phase 2, so you don't have to wait for production. The moment you get approval by FDA, you go into production and selling the compound.
Great. I think that's really interesting for me, not being in this field, hearing that timeline and all the different steps that have to happen. There's a few questions also about people potentially wanting to volunteer for clinical trials, and how they can be involved. And I don't know if you have any information on that, since it is a little bit further out.
Well, thank you for volunteering. As I mentioned, so the first, you see, when there's a new medication, the first clinical trial is going to be with the simplest indication. So a simplest indication in this case is acute Lyme, right? You probably, people are asking this question, probably are not asking it about acute Lyme. So the plan would be to get it approved against acute Lyme. And then I would imagine, even though I'm not advocating for that, but I'm just laying it out how it is, it's the reality. The reality is that once it becomes available, then any doctor can prescribe it for anything, including chronic Lyme. So you do not necessarily have to, wait for a trial for patients with chronic disease. And if anything, I think it would be considerably more sparing to patients to use a selective antibiotic rather than a broad spectrum.
There's a question, if Hydramomycin A was discovered in the 1950s, why has it not been used previously, or was it used for other purposes and not to treat Lyme's?
Yeah, so I was asking myself a somewhat related question when I was astonished looking at the database that we discovered and all that, but after all, our logic I laid out to you. I was thinking, how could they possibly have discovered it? It's because the activity is so poor. They should have missed it. The only way to properly discover it if you test it directly against spirochete. The answer to that question is that the producer of this compound, Streptomyces hygroscopicus, makes it in huge amounts. For whatever reason, they decided that they're going to really zap all the spirochetes in their environment. They don't like spirochetes, right? So then, even though activity is poor, you can pick up that activity if you're testing it against other bacteria. A little bit of activity you can pick up. And that's apparently how it was discovered. But then when people tested against conventional pathogens, it was fairly inactive. And no, it was never used or developed against anything.
There was a question something that I had as well, which was, so moving forward with clinical trials and potential treatment, is this really talking about treatment? So it'd be kind of a continued treatment over time for long Lyme, for example, or would it be a quote unquote permanent immunity or permanent solution?
Well, it is. So one approach would be to restore the microbiome, to fix the microbiome. And there, so we have, I would say, one very good a reference point now that is coming up from Schroeders Therapeutics. That's a company in Cambridge that is developing the first which will probably be the first approved microbiome intervention against the Clostridium difficile infection. And so the therapeutic is a pill that carries symbiotic Clostridia. So they outcompete, the bad guy. That's how you get cured of serious cases of infection. What they found, interestingly, is that the bacteria that they deliver in the pill engraft in the colon of patients with Clostridia difficile. That means that they become normal inhabitants. And so you don't have to, take those pills all your life. You take a course and, the C. difficile is gone, the good guys are in. So that's the kind of optimistic scenario that we're probably looking at. In the case of the antibiotic, well, if you have acute Lyme, you take the antibiotic, Lyme is gone, and you didn't get long Lyme. So that's our hope. And if you do have long Lyme to begin with, and there is residual pathogen, which is unclear. But if, let's say, Hydramomycin A helps, well, it will help by eliminating that residual pathogen. You won't have it anymore.
So there was, so I guess a sort of follow-up question too was, in the absence of that treatment being available for humans now, is restoring the microbiome in general curative and treatment? And then this person is specifically asking about like fecal transplants and fully restoring microbiome.
Right. So, well, I don't know about whether it's curative or not, because we'll only know when we develop a consortium of bacteria we're working on, and then hopefully test it in humans, which will take some time. Now, fecal transplantation is an interesting idea, it's an interesting proposition. We sort of, my first idea was to try fecal transplantation. I even sort of made an arrangement with a company here in Massachusetts that sort of handles those things. But then they started reporting cases of death due to fecal transplantation. So cases, they're not very frequent, but the cases of death were due because so a donor would have, for example, in their stool, would have toxigenic E. coli. So some E. coli carry toxins, right? Some strains of E. coli carry toxins. And if a person is healthy immune system, et cetera, or whatever reasons, the person could be perfectly healthy and not know that they're carrying this E. coli or some other nasty bacteria. And then the recipient gets it and the recipient dies because it starts blooming in the recipient for some reason. And so then FDA kind of started putting a lid on fecal transplantation, apart from strictly approved cases like where there's really no other solution, like Clostridium difficile infection right now. Serious cases of Clostridium difficile infection that are not treatable by antibiotics. So I don't have a good feeling at the moment whether fecal transplantation would even be allowed in such an exploratory. You see, this hypothetical case, I cannot tell the FDA that I know for a fact that fecal transplantation is going to work. I don't know that. So then getting the approval becomes problematic, at least in the US.
So there's a question about messenger RNA technology, and are you optimistic for the possibility of messenger RNA technology to combat tick-borne diseases as well?
Yes, I know. So Charles, my other collaborator, Charles Chu from UCLA is working on this, and I like the results that he's getting.
Excellent. Let's see, we're going to be wrapping up shortly, so I'm going to pick a few more questions. People, COVID is on everyone's mind right now, and how the messenger RNA, vaccines were available, were kind of fast tracked, right? So the question is, understanding that life and death were on a worldwide scale, but for those of us, the person asking this question with Lyme's that are suffering every day, is there a possibility of speeding this up, or how does that get sped up?
That's an excellent question. I was discussing that recently with guys from FlightPath. That's a company that is focused on developing therapies against the Lyme. I think that's the only company that I think is doing that currently. And what came to our attention was not even so much the vaccines that you're mentioning, because vaccines is kind of, you can say it's a separate beast, right? Vaccines are not small molecule drugs. They're in their own category. But the pills, anti-COVID pills that are being developed, by Pfizer, by Merck, by several other companies, the Merck pill. If you look at the approvals that they got from FDA, everything was wrapped up in 1 year, within a year, right? That's astonishing. So maybe the time FDA will start moving its timelines. It's possible.
Thank you. So Louise asks, since Hydramomycin A is active against ribosomes and protein synthesis, is it likely to be effective against Lyme persister forms, which are dormant?
Yeah, that's a good question. I actually happen to be an expert on persisters. All right, that's a hat, if you will. It's directly no, but we recently published an interesting approach of eradicating persisters with antibiotics that do not kill persisters. I hope that makes no sense to you, right? Because it is a bit counterintuitive. So what we did, starting in vitro in a test tube, we developed pulse dosing, and we checked it out on two pathogens, on Borrelia burgdorferi and on Staph aureus. The idea is very simple. So here you have a population of pathogen cells in your test tube, and a small subfraction of that are dormant. They go into dormancy spontaneously. And it's very difficult to impossible to kill them with conventional antibiotics. So you introduce an antibiotic. Excuse me. The antibiotic, like Hydramomycin, kills regular cells. Persisters are alive. Then you emulate what normally happens in the body. Normally in the body will wash out the antibiotic after a while. So you eliminate that antibiotic from population, and now they wake up, persisters wake up and start regrowing. But you don't allow them to go through these divisions and rebuild the population with new dormant cells, right? You hit them with antibiotic again, but they woke up. So they woke up, they became susceptible. They turn on protein synthesis, they're susceptible, so they collapse. Again, you repeat it several times and you get full sterilization with an antibiotic that does not on its own kill persistence, right? So we call that pulse dosing. That I think may be a very viable way, if there are remaining persisters, to develop a pulse dosing regimen with Hydramomycin A and get rid of everything, including dormant cells.
Thank you. Dr. Lewis, I think we are going to start to wrap up. It's a little after 6 now. For those who joined late, we had a few people say they joined late and so had some questions. We are recording this, or we have recorded Dr. Lewis's presentation today, and we will be posting that on the Linda Loring Nature Foundation YouTube channel, which is linked to our website probably later this week, but we'll send a link to everyone who had registered today. So if you missed anything, if you want to go back to hear something that you kind of caught quickly, then that will be available later this week. I want to thank you so much, Dr. Lewis. I was really fascinated also by hearing how you develop ideas. I think that is an underappreciated thing, and especially academia, not criticizing each other. I mean, that's novel in itself. So I appreciate that.
Thank you, Sarah.
Well, thank you again for joining us, and we hope to see you all again at a future Science Pub.